Hi, I'm Tumi. I'm a graduate student in Alex Kulikov's lab at Cold Spring Harbor Laboratory. You have heard from some of the talks about algorithms to represent visual stimulus. Today, I'm going to switch gear and tell you about our work building a model for the olfactory system. So for those who are not familiar, this video will give you an overview of what happened when we smell something. So we get cues from the environment around us, not just through seeing, but also through smelling odorant molecules in the air. These molecules fly into our nose and interact with neurons there. These neurons have receptors on the cell membrane, which help transduce chemical signals in the environment to electrical signals in the brain. You may notice that there are many different colors here of the neurons, and that's because in our genome, there are 350 different genes that code for olfactory receptors, and each cell chooses one of these genes to express. The neural signal is then carried further into other regions of the brain and is processed in ways we don't fully understand yet. But to sum up, you have a chain of events from molecules to receptors to neural activity, then to perception. So just to reiterate in this picture here, olfactory receptors are large molecules on the membrane of the cell that help convey information about the structure of the molecules and lead to perception of the odors. We know that these receptors care about the three-dimensional structure of the molecules and that they are chiral because molecules that are mirror images and are not superimposable smell differently, and I will give some example later in the talk. We also know that given an odorant molecules, there are many possible conformations and orientations, and only one or a few of them are relevant as they are the actual conformations that bind to the receptors. So this is one place where the olfactory system differs from the visual system. There's one copy of an image at a given time, but for odors, there are hundreds of copies of an odorant, each with different orientations and conformations interacting with the receptor at once. Therefore, we need to consider these factors when we develop a model for odorant recognition. So given the considerations that I told you previously, we decide to present an odorant molecule as a 5-dimensional tensor. So you have 3 dimensions for space, 1 dimension for the features vector. So just like in images, you have different color, the RGB channels. In this case, an atom can be either carbon or hydrogen or chloride. And then the last dimension is for different orientation and conformation of the molecules. So in this example, you see this molecule chlorobenzene. It has 6 carbon atoms. So you would see 6 3D cloud in this carbon channel. And you have 5 hydrogen in the hydrogen channel and etc. And we can rotate this molecule to obtain different orientation. So I mentioned previously that to recognize odorant in the environment, we, in human, we have an ensemble of 300 olfactory receptors. And we want to understand what factors influence the composition of these olfactory receptors and how they evolve. So our strategy is to use conventional machine learning technique such as backpropagation and convolutional filters to train this and then to train this network so that we can approximate the responses of these receptors to odorant stimuli. So as you might know, training these convolutional neural networks require a lot of data. For smell perception, the data set that we have is on the order thousands of label molecules. And by label, I mean you have a molecule and it has the percept associated with it. So floral, fruity, minty, sulfurous, etc. but there are a lot more molecules that we don't have these odor annotations for. These are the molecules on the left where we have access to their three-dimensional structural information, but we don't know anything about how they smell. So our strategy is to implement 2 stages of training, taking advantages of both of these source of data. In the first step, we would use unsupervised learning to obtain a representation of the odorant chemical space. And then next, we use supervised learning to predict how this odorant would smell. So like I said, in the first day, we would train an odorant encoder, and we name this network DeepNose. This encoder would transform the molecule input, which was a 5-dimensional tensor that I said previously, into a latent representation with 50-fold fewer units, from which the decoder had to reconstruct the input. And it consists of many 3-dimensional convolutional layers. Since the encoder's job is to extract the most salient structural features of a molecule, we think it is functionally analogous to the responses of the ensemble of the 350 olfactory receptors. So can we do that? The answer is yes. We were able to train autoencoder on these molecules. We evaluate the autoencoder by calculating the correlation coefficient between the input and the reconstruction for molecules in the test set. And as you can see, this network is capable of reconstructing the molecule accurately as a whole, but also do it very well for each of the features. So all the elements, it was able to do that. Just to show you what it looks like. So I'm showing how the reconstruction of the test set improved as we continue to train the autoencoder. The top row show the 2D projection of the input molecule. So you would see, I separated by the 6 channels, so you could see the bottom show the reconstructed molecules. Let me see if I can play this again. And at the beginning, you see that the reconstruction is not very good, but towards the end, we were able to match the input accurately. So we have this autoencoder, and next we want to train a classifier on label molecules. What we did is we transfer the train encoder's weight to the classifier and pair it with fully connected feed-forward neural networks to predict smell percept. And just a little bit more about the smell percept data set that we're using. I'm talking about the data set that I'm presenting is called the Good Sense data set, and I list the website here for those of you who are interested. This data set has over 3,500 odors with 400 words. So each molecule is associated with one or more semantic descriptors, such as floral or fruity. For this data set, it's 0 or 1, but you could also have different data sets that have a graded responses. I'm showing 2 example data points on the right. If you're thinking to yourself that these molecules look similar, you're correct. In fact, they are mirror images of each other. One is R-carbon and the other one is S-carbon, and they have different smells. So just to reiterate the point that the nose is chiral, and that's why we chose a representation that we did. So I trained the classifier on these descriptors, and I evaluate the performance of this classifier using the area under the ROC curve. The score close to 1 means this classifier can perfectly separate molecules with the descriptors from those without the descriptors. And a value of 0.5 means it's a random classifier. We see that there are many descriptors, but some descriptors are better predicted than others, such as fishy, garlic, sandalwood. But overall, this performance is comparable with other molecular encoding methods. next, we are interested to interpret the prediction of this DeepNose classifier. We want to find which part of molecule would give rise to certain prediction. To do this, we borrow this technique that was developed to interpret classification of visual stimulus, and it's called occlusion analysis. The idea is very simple. You just occlude a region of the image and see how it affects prediction. And they were used to identify pixels responsible for the dog in the first image and the car wheel in the second image. So similarly, for DeepNose, I would apply this technique. In this case, we would occlude pixels associated with an atom and see how it affects the prediction score. In this case, I have this molecule, trimethyltryptamine. This is a notoriously fishy molecule. This is one molecule, but there are two coloring schemes. On the left, I color by the atom identity. So black for carbon, blue for nitrogen, white for hydrogen. And on the right, I color according to the contribution of the atom to the percept. So this result confirm what we have known. So it showed that the presence of the nitrogen atom make the molecule makes the molecule more fishy. I'm gonna show you a couple more examples. We have here two fruity molecules. We see that for both of the molecules, the top and bottom, the atom involved in an ester group is associated with fruity scent. And one last example is the cinnamon odors. And it's very striking that the atom associated with benzene rings contributes to this classification. So it's gonna wrap up my talk. We use neural networks to model interaction of olfactory receptors with molecules, and we view olfactory receptors as three-dimensional convolutional filters that analyze molecular shape, and we want to use this network to understand the constraint on the evolution of these receptors. With that, I want to thank my lab. I'm in the Alex Kulikov's lab, and my collaborators in this project are Sergey Shuvaev, who gave a talk yesterday, and Daniel Keppel. And you can check out these papers if you're not interested. Okay.
Awesome. Thank you so much, Tumi. We've got a couple of questions here and a little bit of time for them. So the first is from Victoria. Thank you for the fascinating topic. You said that there is up to 30K genes coding the perception of an odor. My question is, can we even rely on the annotated part of the data, even the unique receptor configuration? The perception of the odor is up to genetic encoding.
We have 350 genes. That's in human, and in mice we have around 1,000.
Okay.
I don't.
Yeah. Where the 30K came from? Yeah, I'm not sure, but you don't know that.
The annotated part of the data. Yeah, I'm not sure I understand the question.
Okay, maybe we can come back.
Maybe we can chat later.
Yeah, it would be great. Okay. So the next one is very interesting approach. Can you comment on the similarity of your approach to biological network structure? The encoder in biology doesn't seem to be very deep. ORNs project a bulb and then piriform / olfactory cortex in mammals, so only a few layers. And some people think the representation in piriform is a nonlinear random projection. Curious to hear whether you've tried those kinds of simple networks as well.
Right. So I guess when you talk about the recurrent connection, I guess it would be more relevant to how I approximate the classifier. But if you're talking about the encoder layer that use a series of three-dimensional convolutional network, yes, like it is using many layers. But for the encoder part, we're trying to approximate the interaction between the molecule and the receptors. You can imagine that each of these receptors are amino acid that interact with small parts of the molecule. And as you have more layers, you would have more long-range interaction of these amino acid. But yes, the classifier is more simplistic, using a feed-forward layer to approximate is our effort to try to simplify this part that we actually don't really understand. The recurrent.
One last question. Do the basis learned from the autoencoder have response properties similar to olfactory receptors? How many basis elements do you need to reconstruct molecules?
That's a very interesting question, and that is something that many people have tried to investigate. The problem is we don't have enough experimental data. So a group like Joel Manlin have tried to, probe the responses of olfactory receptors to a few hundred odorants, but I think it's actually less. But yeah, like we are. This is one of the future direction. Once we have data from our collaborators such as Florin at Cold Spring Harbor, and I think Dima Rinberg at NYU is also working on this. We would be interested to compare with experimental data.
Cool, great. Okay, thank you very much, Tumi.
Okay.
Great talk, and we're gonna move on to I think the last. Where are we at? I think that perhaps the last speaker of this session.